All right, so you started the basic notes for Hardy-Weinberg Equilibrium on Tuesday. So now we're going to start the lab, and this is actually going to be a modeling lab. I'm not going to complete it for you as a demo, but we're actually going to complete it together. I'm going to kind of guide you through some of the steps, and then you're going to make some exploration on your own. So today is a heavy guidance day, so we're going to kind of work at the same pace and pause this video whenever you need to, or rewatch certain parts to make sure that you got all of the necessary instructions. Obviously, if you're good at reading, you can just follow along the PDF of the lab as I have open on the left side over here, the same way that you would just read through and follow instructions. If you want to, you don't actually have to watch this video. But before you log off, if you're planning on just reading it, make sure you get up to all of the steps on page 12 or S36 before you screenshot for today and submit that in the post below this one. If you want to follow along, we're going to walk through the background and then we'll talk through the steps on how to set up a basic premise for one generation of one population in Hardy-Weinberg Equilibrium. And that's going to set you up to be able to kind of work on your own at the start of next week to pull different ratios and different generations on your own. That way I don't have the same thing from everybody who's just kind of following what I'm working on. So the title of the lab is Mathematical Modeling Hardy-Weinberg. And the reason we can model it is because when we do practice problems, you'll see basically Hardy-Weinberg Equilibrium is just based on like p squared plus 2pq plus q squared equals 1. So it's like the quadratic base formula. And it always follows those rules, so we can do calculations pretty easily with it. And so we're going to use a Google spreadsheet in order to model this. Usually we would use Excel because there's more functions on Excel and it's a little bit easier to work with. But you're actually going to open up a Google spreadsheet. And you can do that by going to your Google Chrome, opening up a new tab. Let me move myself. So when you open up a new tab, by your username, when you're logged in, you should have this grid shape for Google Apps. And when you click on that, there's a Google Sheets, and you just click on New and Blank. We're definitely going to name this so that when you turn it in or share it with me, I can actually view it and grade it. And so what you would want to name it is after the lab, right? Hardy-Weinberg. And then you're going to want to do an underscore and then put your name. I only have this one class period of AP Bio, so you don't have to worry about your class period, but put your name at the end so that I know whose assignment it is when you share it with me and I actually get to grade it. Okay, so let's leave that aside for now. We're going to just go through the PDF. You can have yours open on your screen too for more details. We're just going to go over the background. So it's asking, how can mathematical models be used to investigate the relationship between allele frequencies in populations of organisms and evolutionary change? Well, evolution occurs in populations of organisms and involves variation in the population, heredity, and differential survival. One way to study evolution is to study how frequency of alleles in a population changes from generation to generation. In other words, you can ask, what are the inheritance patterns of alleles, not just from two parental organisms, but also in a population? You can then explore how allele frequencies change in populations and how these changes might predict what will happen to a population in the future. Mathematical models and computer simulations are tools used to explore the complexity of biological systems that might otherwise be difficult or impossible to study. Several models can be applied to questions about evolution. In this investigation, you will build a spreadsheet that models how a hypothetical gene pool changes from one generation to the next. This model will let you explore parameters that affect allele frequencies such as selection, mutation, and migration. The second part of the investigation asks you to generate your own questions regarding the evolution of alleles in a population. Then you are asked to explore possible answers to those questions by applying more sophisticated computer models. These models are available for free. We won't actually do that part. This investigation should also provides an opportunity for you to review concepts you might have studied previously, including natural selection as the major mechanism of evolution, the relationship among genotype, phenotype, and natural selection, and fundamentals of classic Mendelian genetics. So we just did Mendelian genetics when we did Punnett squares, codominant and complete dominance, multiple alleles. We have talked about genotypes and phenotypes. Natural selection we'll get into more detail in one of our upcoming units. There aren't really any safety precautions. So Getting started. This particular investigation provides a lab environment, guidance, and a problem designed to help you understand and develop the skill of modeling biological phenomena with computers. There are dozens of computer models already built and available for free. The idea for this lab is for you to build your own from scratch. To obtain maximum benefit from this exercise, you should not do too much background preparation as you build your model and explore it. You should develop a more thorough understanding of how genes behave in a population. To help you begin, you might want to work with physical models of population genetics, such as simulations that your teacher can share with you. With these pencil and paper simulations, you can obtain some insights that may help you develop your computer model. So we're actually going to just do the computer model straight away. And the procedure is as follows. It is easy to understand how microscopes open up an entire new world of biological understanding. For some, it is not as easy to see the value of mathematics to the study of biology. But like the microscope, math and computers provide tools to explore the complexity of biology and biological systems, providing deeper insights and understanding of what makes living systems work. To explore how allele frequencies change in populations of organisms, you will first build a computer spreadsheet that models the change in a hypothetical gene pool from one generation to the next. You need a basic familiarity with spreadsheet operations to complete this lab successfully. You may have taken a course that introduced you to spreadsheets before. If so, that will be helpful, and you may want to try to design and build your model on your own after establishing some guidelines and assumptions. Otherwise, you may need more specific guidance from your teacher. You can use almost any spreadsheet program available, including Google Sheets, to complete the first section of your investigation. In the second part of your investigation, you will use more sophisticated spreadsheet models or computer models to explore various aspects of evolution and alleles in populations. To understand how the complex models work and their limitations, you first need to build a model of your own. So we are going to go ahead and build a simple mathematical model. And the real world is infinitely complicated. To penetrate the complexity using model building, you must learn how to make reasonable, simplifying assumptions about complex processes. That way we don't have to work with a bunch of rules, and we'll work with some broader rules that cover everything. For example, climate change models or weather forecasting models are simplifications of very complex processes, more than can be accounted for with even the most powerful computer. These models allow us to make predictions and test hypotheses about climate change and weather. By definition, any model is a simplification of the real world. For that reason, you need to constantly evaluate the assumptions you make as you build a model, as well as evaluate the results of the model with a critical eye. This is actually one of the powerful benefits of a model. It forces you to think deeply about an idea. There are many approaches to model building. In their book on mathematical model building in biology, Otto and Day in 2007 suggest the following steps. 1, to formulate the question. 2, determine the basic ingredients. 3, qualitatively describe the biological system. 4, quantitatively describe the biological system. 5, analyze the equations. 6, perform checks and balances. And 7, relate the results back to the question. So as you work through the next section, record your thoughts, assumptions, and strategies on modeling in your laboratory notebook. So always make notes. If you want to, you can do it on the side of this PDF if you've already saved it. Step 1, formulate the question. Think about a recessive Mendelian trait, such as cystic fibrosis. Why do recessive alleles like cystic fibrosis stay in the human population? Why don't they gradually disappear? Now, think about a dominant Mendelian trait, such as polydactyly, or having more than 5 fingers on a single hand or toes on a foot in humans. Polydactyly is a dominant trait, but why is it not a common trait in most human populations? How do inheritance patterns or allele frequencies change in a population? Our investigation begins with an exploration of answers to these simple questions. So step 2 would be to determine the basic ingredients. Let's try to simplify the question. How do inheritance patterns or allele frequencies change in a population? With some basic assumptions. For this model, assume that all the organisms in our hypothetical population are diploid. This organism has a gene locus with 2 alleles, A and B. We could use A and lowercase a to represent the alleles, but A and B are easier to work with with the spreadsheet you'll be developing. So technically, A will represent capital A and B will represent little a. Okay, they're going to be for the same allele, but when you're writing on spreadsheets, they automatically capitalize the first letter. So if you leave like a small a, it's going to turn into a capital A, and then you'll get really confused. So far, this imaginary population is much like any sexually reproducing population. How else can you simplify the question now? Consider the population has an infinite gene pool, aka all the alleles in the population at this particular locus. So gametes for the next generation are selected totally at random. What does that mean? Focus on answering the question in your lab notebook for a moment. It is a key to our model. For now, let's consider that our model is going to look only at how allele frequencies might change from generation to generation. To do that, we need to describe the system. So we are going to qualitatively describe the system now in step 3. Imagine for a minute the life cycle of our hypothetical organism, C. If you draw a diagram of the cycle, be sure to include the life cycles of the organism. Your life cycle might look like figure 1. To make this initial exploration into a model of inheritance patterns in a population, you need to make some important assumptions. That all the gametes go into one infinite pool, and that all of them have an equal chance of taking part in fertilization or formation of a zygote. For now, all zygotes will live to be juveniles, and all juveniles will live to be adults. And then no individuals enter or leave the population, right? No migration. There is also no mutation. Make sure to record these assumptions in your notebook. Later, you will need to explore how your model responds as you modify or change these assumptions. The reason why we made these assumptions is because we're going to be maintaining Hardy-Weinberg equilibrium, at least for the initial stages. Okay, so again, no migration, no mutations, all of the above are going to prevent allele frequency changes that are happening outside of our normal generation and inheritance. And then also, obviously, these, what does it say? Gametes being equally likely to be fertilized, obviously no preferential mating. Step 4 is to quantitatively describe the biological system. Spreadsheets are valuable tools that allow us to ask what-if questions. They can repeatedly make a calculation based on the results of another calculation, and they can also model the randomness of everyday events. Our goal is to model how allele frequencies change through one life cycle of this imaginary population in the spreadsheet. Use the diagram in figure 1 as a guide to help you design the sequence and nature of your spreadsheet calculation. The first step is to randomly draw gametes from the gene pool to form a number of zygotes that will make up the next generation. Keyword random. To begin this model, let's define a couple of variables. Let P equal the frequency of the A allele, and let Q equal the frequency of the B allele. So you're going to bring up the spreadsheet on your computer. The examples here are based on Microsoft Excel, but any modern spreadsheet can work, including Google's online Google Sheets and Zoho's online spreadsheet. Hint, if you are familiar with spreadsheets, the randomizing function and using the if conditional statements to create formulas in spreadsheets, you may want to skip ahead and try to build a model on your own. If these are not familiar to you, proceed with the following tutorial. Somewhere in the upper left corner, in this case, cell D2, enter a value for the frequency of the A allele. So it's in D2 because we're supposed to write out what they wrote earlier in the let section. So let's just write it out to make it easy for us to reference. So we're going to write in our spreadsheets P equals the frequency of the capital A allele. And then we'll also write in line 2 that lowercase q equals the frequency of the capital B allele. And if you're bad at typing like me, it's going to get exposed in this video. Okay, so somewhere in the upper left corner, in this case cell D2, we're going to use D2 as well since it's a little bit away from the facts that we just wrote in the upper right hand corner. Enter a value for the frequency of the A allele. This value should be between 0 and 1 because all percentages are between 0 and 1, right? Because 0 represents 0% and 1 represents 100%. So anything in between 0 and 1 is going to represent a certain ratio or a certain percentage of chance. Go ahead and type the labels in your other cells, and if you wish, shade the cells as well. This blue area will represent the gene pool for your model. Highlight the area you wish to format with color and right-click with your mouse in Excel to format. This is a spreadsheet, so you can enter the value for the frequency of the B allele. However, when making a model, it is best to have the spreadsheet do as many of the calculations as possible. In all of the alleles in the gene pool, all of the alleles in the gene pool are either A or B. Therefore, P plus Q equals 1, and 1 minus P equals Q. In cell D3, enter the formula to calculate the value of Q, which is going to be, in spreadsheet lingo, 1 minus D2. So the way this works is we're going to want to write, here we're going to want to write P equals, and underneath it we're going to want to write Q equals. And over here we're going to pick a number. Any number between 0 and 1, okay? In this tutorial where I'm guiding you, you can pick a number that is the same as mine, or you can pick a different number. That is up to you, okay? The whole thing will work out the same, whether or not you pick the same number or a different number, but you need to just follow the same steps, okay? So your number doesn't have to be the same as mine, but you can have the same number if you wish to for now. When you do your own work, you are going to have to change your numbers. Okay, so the way that we're going to automatically calculate Q and not have to use our brains at all And make sure that our spreadsheet always stays regulated is going to be by typing in the formula that is shown at the bottom here, which is going to be 1 minus D2. So you have to write the equals because that indicates to the spreadsheet that you're going to enter a function. And it's going to be 1 minus, and then you will just click on D2. And if you notice in my formula box right here, the formula that is going to be going into this box for what Q equals is going to equal 1 minus D2, which is 1 minus the p-value, which is 1 minus 0.24 in this case, and automatically it's already calculated for you. You don't need to click on it. You would just click enter, and it will automatically enter it. Now, the key way for me to tell if you did this right when I'm checking your work is if I click on your box and look up here to the formula box and it just says 0.76 because you manually did the math and you typed it out, then you're not going to be able to get the right number whenever you automatically adjust, right? So if I were to change this number to 0.36, then once I click enter, anything that is involved with D2 will automatically change as well. So if I change this to 0.36, the Q automatically changes to 0.64 because there's a formula in here instead of a value. That way I don't have to manually go type it in. If I want to change this to the ever so popular 0.5, then the Q value also automatically changes to 0.5. So it's super, super convenient when we use formulas like this. I'm gonna go back to my original value just because I like this number. Okie dokie. Next step now. So our spreadsheet should look something like figure 2. You can highlight if you want to. You can change different colors if you want to. It's not really important to do that. Change all of them, otherwise it's kind of ugly. So you can highlight that if you want to. You can highlight this part if you want to. Nobody really, it's not a huge deal. Okay. So let's explore how one important spreadsheet function works before we incorporate it into our model. In a nearby empty cell, enter the function and then we will remove it later. Okay, so you're going to do some practicing about, and you will just close it. Okay, so this function is the randomizing function. And the way it works is that when you just enter the equal sign, obviously you're getting ready to tell the formula box that you are going to put in a function or a formula, and the function or formula that you're putting in is the randomizing function. Now, whenever you click it and you don't give it any number ranges inside of it, the automatic range that it's going to give, look, it says returns a random number between 0 inclusive and 1 exclusive. So it's going to give you a number from 0 to anything else that's under 1. So it could go 0.999999999, but it's random, so it's likely that you might never see any of the any number like that. You can, every time you click enter or every time you click on the box, it'll give you a different number. I think. Hold on. Let me read through this again. So let's explore how one important function works. Note that the parentheses have nothing between them. After hitting return, what do you find in the cell? If you're on a PC, try hitting the F9 key several times to force a recalculation. So I think you have to kickstart the recalculation with F9. Let's see. I'm not exactly sure. Seems like this one's... So if you're on a PC, try hitting the F9 key several times to force recalculation. On a Mac, enter CMD or CMD plus or CMD equals. What happens to the value in the cell? Describe your results in your lab notebook. So the way the randomizing function works is that it returns you a random number between 0 and 1 in a decimal format. This is a powerful feature of spreadsheets. It allows us to enter a sense of randomness to our calculations if it's appropriate. And here it is when we are randomly choosing gametes from a gene pool. So go ahead and delete the random function in the cell. Let's select 2 gametes from the gene pool. In cell E5, let's generate a random number, compare it to the value of P, and then place either an A gamete or a B gamete in the cell. We'll need 2 functions to do this, the randomizing function and the if function. Okay, so we're going to go ahead and start using the randomizing function. So we're actually going to delete this one now, and we're going to start working in the E5 column. E5 is down here. And so the function that we want to enter in E5 is the function shown where it says E equals, and then we're going to use if, and then we want to parentheses so that it's encompassing it. So we're going to use the randomizing function with a parentheses. If if this random number is less than or equal to $2, then we're going to ask them to drop an A in the box. And if it's not, we're going to ask them to drop a B in the box. So let me go through exactly what every single symbol in here means. So basically the statement is saying in box E5, if the random number generator number that is produced is less than or equal to the p-value that we established in D2, then the function is going to put a letter A in this box. So it means that if it's a 0.13, you're going to see an A. If it's a 0, you're going to see an A. If it's a 0.2399999, you're going to see an A. However, if it's greater than a 0.24, so if you get a 0.572, it's going to be a B in the box. Okay. And so once you click enter, you would get an A, right? And the reason we put a $sign is that every single time, technically, when you take this little thing and you drag it down, it'll do the function again for you, and it'll re-randomize it every single time. The problem is that if you did this and you didn't put the $sign next to the D, between the D and the 2, every time you shifted this down 1, it would shift the reference box down 1. So instead of referencing this box, it would start referencing D3 and D4 and D5 every time you pulled it down 1, 2, 3, 4, 5, right? We don't want that to happen because we only ever want to reference the p-value that we set as the frequency for the dominant allele. So we're going to make sure that the $sign is between the D and the 2 because it locks in the box that we're referencing. Okay, so like I mentioned earlier, there's this cool little box that's on the bottom corner of every single box that you have a function in. And if you take it, it does this magical thing where it drags and copies the exact same function into the box underneath, to the right, to the left. So only when you put your cursor over this little dot, it's going to change from a regular cursor into this plus sign, and then you can drag it down however many times you need to, and it'll give you a bunch of B's or A's depending on with the random number generator. Did it pull a number less than or equal to 0.24, or did it pull a number higher than 0.24? Okay. The formula in the cell basically says that if a random number between 0 and 1 is less than or equal to the value of P, then put an A gamete in this cell. If not, then it would be higher than P, then it's going to put a B gamete. The if functions and the random functions are very powerful tools when you try to build models for biology. Now, create the same formula in cell F5. Make sure that it is formatted exactly like E5. When you have this completed, press the recalculate key to force a recalculation of your spreadsheet. The easiest way this thing recalculates is just by dragging and dropping. It'll recalculate all of them. It forces the whole sheet to recalculate any random value. Okay, so we're going to do an F5, and we're still going to do the exact same formula. So if you want to, you can literally just copy and paste it, or you can retype it out. That is up to you. When you copy and paste it though, double check all of your values. Sometimes this will shift. It might shift over one, and you don't want that to happen. So you got to always double check if you're planning copy and pasting. Does it still say if parentheses random parentheses less than or equal to $2? You put an A. If it's greater than, you would put a B. Okay, that's correct. Try recalculating 10 to 20 times. So we're going to drag it. How many times did I drag it? 1, 2, 3, 4, 5, 6, 7, 8. I mean, I can't count. So we'll go 21 minus 5. So that's about 16. So between 10 to 20 times, yeah, sure. Are your results consistent with what you expect? Do both E and E5 and F5 change in A and B ratios? Try changing your p-value or your 0.0 to 0.8 or 0.9 and see what happens. So if we change this to 0.8, oh, why is this font all weird? It's kind of funny. Okay, so let me change the font back. I don't know why the font looks like this. It's kind of ugly. Okay, so if you change the values here, it automatically changes here because guess what? This is tied to this because D2's value is inside of this formula. So what's nice about the spreadsheet is that these formulas that are using reference boxes automatically change for you. An easy way for you to label these would be just to write gametes so that you know what we're referencing down here. I think pretty much every time you press enter on the spreadsheet inside of a box, it'll change it. Okay, you could stop here and just have the computer recalculate over and over, similar to tossing a coin. However, with just a few more steps, you can have a model that will create a smaller number or a larger number of gametes for the next generation. Count the different genotypes of the zygotes and graph the results. So you're going to have these two formulas in E5 and F5 down, and we're going to go about 16 rows to represent gametes that will form 16 offspring for the next generation, as in figure 4. So we're kind of already there since we did the drag and drop. So to copy the formula, click the bottom right-hand corner of the cell and your finger press down on the mouse, drag the cell downwards. You're going to click and drag that little box right here, or sorry, that little dot on the corner of the box, and you will drag it down. And if you want to, you can put a border so that you can differentiate the gametes in lower. You can change colors if you wish to, like they did. That's not really super important. I'm not going to grade you for or against if you do that. Some of my kids in my last years, they actually color coded every single generation by a different color, and it was pretty cool, but it doesn't help or erase from the math, just so you know. Okay, so we will put the zygotes in cell G5. The zygote is a combination of the two randomly selected gametes. In spreadsheet vernacular, or in spreadsheet language, you want to concatenate the values in the two cells. In cell G5, enter the function equals concatenate E5 and F5, and then copy this formula down as far as you have gametes on, as in on figure 5 on the next page. So basically, we're going to take the formula concatenate, and we're going to start in G over here, and we're going to write equals. So you can already see that there's a concatenate, and then you're going to put a parentheses E5 and F5, and then you're going to close the parentheses. And it shows you by highlighting in color which boxes you're referencing here. So if you concatenate them, then it shows you that it pulled these two and put them together for you. That's it. The concatenate is literally just in charge of putting them together. And it's kind of a convenient tool because that way you can combine what's in two boxes into one in a nice orderly format. And we're going to click and drag because we're lazy and we don't want to rewrite every single formula, right? So if you want to double check the computer, they're probably not going to make mistakes, but you can visually check it just to understand it better. AA is pulled together here. AB is pulled together here. BB is pulled together here. AB is pulled together here. It's pretty cool. Okay. And we're going to want to name over here. Let's put a border again, and we want to tell that it's the zygote. Okay. And we can color code this one as well, like they did. So we have the zygote. The next column on the sheet, H, I, and J are used for bookkeeping. That is keeping track of the numbers of each zygote's genotype. They are rather complex functions that use if functions to help us count the different genotypes of these zygotes. So the function in cell H is going to be equals if G5 equals AA, then 1, 0, which basically means that if the value in G5 is AA, then put a 1 in this cell. If not, put a 0. Okay, and then we're also going to do a very similar function in cell J5, but in H5 right now. Oh, one thing I want to bring up. So when you notice when we wrote the formula for the zygote, we didn't put dollar signs between the E's and the F's because in this case, whenever we dragged it down, we did want the reference frame to drag down too. So if you notice here, it's E6, F6. Here it's E7, F7. So the rows are actually matching up with the rows that the formula is written in because we didn't put the dollar sign there. So it's not actually stuck on one single box. And that's a good thing because we want to reference each row each time we're pulling to the zygote. Otherwise, it wouldn't make any sense to only pull from A and B every single time. Okay, so in a function, the function for H5 is going to be equals the if function G5. Again, we're not using a dollar sign. If it is A, comma A, sorry, capitalize that one. Then put a 1. If not, put a 0. Oh, sorry, there was no comma in between. That's why it didn't work. Okay, so when you click the enter, the randomizing function will re-randomize. But because it's a BA, there's a 0. Okay, and you would drag this all the way down. And every time there's an AA in the zygotes, then you would drag, it would put a 0, a 1 automatically. And every time it's not an AA, it will put a 0 automatically. And that's pretty cool because basically it's helping you count up how many of each type of zygote you have. Okay, so we're going to enter a similar function into the J column now. And so in J5, we're going to write out the formula if in G5 we have the value equals BB, then drop a 1. If not, drop a 0. So obviously, because it's a 1 for AA, it's not going to be a BB, right? So you're going to pull that all the way down, and every time there's a BB, then you're going to have a 1. Okay? And we're going to label these up top. This is for the AA, this is for the BB. And over here, we're going to have the ABs. We're going to drop that border to kind of keep it all organized. Now, the nested if function is very, very important here for ABs because technically in biology, when there's a BA or an AB, they're the same thing, right? If you're writing a capital A, lowercase a, or lowercase a, capital A, it's always referenced as capital A, lowercase a, no matter what the order the gametes come in, right? So the heterozygotes are kind of weird in that we have to nest or put an if function inside of another if function. So that's nesting. So let's tackle the nested if function. This is needed to test for either AB or BA. In cell I5, enter the nested function as below. So there's an extra set of parentheses which is necessary to nest functions. This function basically says that if the value in cell G5 is exactly equal to AB, then put a 1. If not, then if the value in the cell is exactly BA, put a 1. If it is neither, then put a 0 in this cell. So that's what this nested function is saying. It says if it's A, B, or BA, same thing, it's going to put a 1. Otherwise, it's going to put a 0. So you have to make sure that you are putting these functions in exactly correctly. And an easy way that Excel will tell you it's right or it's wrong is if it works or not. And so notice that instead of putting a 0 here, normally we would be like, oh, if it's not an AB, then you're going to put a 0. But we're going to include also that it could also be a BA with this nested function, right? So if G5 is equal to BA in this case, then you would put a 1, if not, put a 0. And there are 3 parentheses to close there. So if you make any mistakes, it shows up as red, which is also pretty convenient. But you want to double check your grammar here in your sentence, right? So it's saying if in the box G5 you have an AB, then write a 1. And if box G5 is a BA, you write a 1. If it's not that, then you would write a 0. So it's having you check for an AB first before it checks for a BA. And if it's neither, then at the very end, it would put a 0. And the suggested autofill is correct. We are going to take that and drag it all the way down. Okay, so we're going to copy these formulas for all the rows that we've produced gametes, and we have it vertically copied all the way down. Enter the labels for the columns you've been working on. So the gametes are in E4, the zygote is in G5 for AA, AB, and BB. Now we have all of our labels done, which is really nice and really nicely organized. So as before, try recalculating a number of times to make sure everything is working as expected. What is expected though? If you aren't sure yet, keep this question in mind as you complete the sheet. You could use a p-value of 0.5 and you'd see your number similar to the ratios you would get from flipping 2 coins at once. Don't go on until you are sure the spreadsheet is making correct calculations. Try out different values for p. Make sure that the number of zygotes adds up. Describe your thinking and procedure for checking the spreadsheet in your lab notebook. How do you check if there are number mistakes here, right? What if, even though it says AA, there's a 0 in AA, and then there is an 1 in AB? What are you supposed to do then? What did you mess up in that case? Right? You'd have to be able to catch those things. Now we're going to copy the cells E5 through J5 down for as many zygotes as you'd like in the first generation. So we went down to around 16, right? Let's first, just as a interesting point to bring up, let's adjust our p-value back to 0.24. So that would adjust our Q value, that would adjust our gametes, and that would adjust our zygotes, and then that would adjust our AA, AB, and BB ratios. So we're going to use the sum function now. So use the sum function to calculate the numbers of each genotype in the H, I, and J columns. Use the genotype frequencies to calculate the new allele frequencies and to calculate new P and Q values. And then you're going to make a bar graph of the genotype using the chart tool. So your spreadsheet should resemble figure 8, and this is where we're going to stop. Okay, so over here, since this is the last individual in the generation that we're making, we'll actually draw a line underneath as well to keep it clarified. And then we're going to want to sum. And sum is an easy function to do. You're just going to write equals, and then you're going to write sum. And you can actually just go ahead and highlight exactly what you want to sum. Oops. Sorry, I need to put a parentheses first. And then you can highlight all the boxes that you want to sum together and close your parentheses and click enter. Every time you click enter, like I said, the randomizing function just re-goes again. So you'd want to sum, and again, don't make my mistake, put a parentheses, and then you can just highlight all of the boxes that you want to sum, parentheses. And like earlier, you noticed if when I didn't put the parentheses, it just turns into an error, and that's an easy way to check that you made a mistake. So this is automatically summing for us how many AAs there are, how many ABs there are, and how many BBs there are. Okay? So if you, let me zoom in a little, and kind of show you what they're playing over here. So what they're showing you is the sum for each genotype, right? This row is going to be the sum for each genotype, and we can write that here. They've written it. I don't know why they use such weird arrows. But using this, then we should be able to figure out how many A's there are, how many B's there are, how many of each allele there is. And so we're going to want to leave some space to do that. And so let's find out how many A's there are and how many B's there are. And the way that's going to work is that we should be able to do some pretty simple calculations. I'm just trying to copy their format, and it's kind of a hassle at this point. You guys don't have to do the lines like I am, the borders. Okay, so the easiest way to calculate this, number of alleles. It's going to be in this row. So how many A's are there? Well, I'm not going to actually manually count because we haven't been manually counting. If you manually count now at this late stage, you're going to mess everything up because remember, almost every single time you click enter somewhere, the randomizing function just kicks off again. So you don't want to have the randomizing function kick off and your number not move with it. So we're still going to be using references and formulas. Okay, so how many A's are there? Well, we know that this genotype has 2 A's in it, and we know this genotype has 1 A in it, and then we know that this genotype has no A's in it. So if we're just looking at A's, we don't really need to consider this one. We just need to consider this one, right? So if we're looking at the sum, we would be writing a formula where we have, how many A's are here? 2. So we're going to have 2 times whatever is in this box. And then we're going to add in, do we need to multiply this one? How many A's are in here? There's only 1, right? So we just directly add this number. Sorry, I don't know why this is equals. You have to add a plus, right? So if you just look at this, we're going to add the 1A from here and 2 of the A's from here. So if there are 2 double A's, technically there's 4 A's, right? And if there's 7 AB's, technically there's 7 A's. And that's what you're going to add together. And so the, what did I do wrong? Oh, hold on. They don't recognize this as multiplication, I think. So you're going to make sure that You're careful with your formulas, and the star is actually the multiplication symbol. So 2 times the AAs over here, plus the singular A's from the ABs. And this should work. So the number is 4 because we have 2 times 0 plus 1 times 4, right? We want to do the same thing with the B's, though, because we want to calculate the B's as well. So you're going to have, for the BB, again, you have 2, right? So we're going to 2, and then we use the star to represent multiplication. 2 times whatever is in this column as the sum of the BBs, plus the singular B that will come from the ABs. And so we have 24. And you can double check this number right now, right? 2 times 2 is 4, plus 6 is 10. 2 times 9 is 18, plus 6 is 24. And as a total, in this population, there are 34 alleles. That means technically we have 17 rows. My bad. Okie dokie. So now we're going to do allele frequency. And the frequency should be pretty easy for you to mentally figure out. So we're going to look at P, and then we're going to look at Q. And since I'm following this, I might as well put borders under it, but it's a pain in my butt. Okay, so for P and Q, we're going to calculate that based off of the A and the B. And the A and the B are based off these. And these ones are based off these. And these ones are based off this. And this one is based off these. And these ones are based off of these, right? So this all is kind of like a waterfall cascading effect. So if anything changes up top with just the p-values, then the whole rest of the bottom flows with a change as well, because it would change this and this and this and then this and then this and then this. Okay, so that's the good thing about spreadsheets. So our p is going to be based on this and this divided by the total number of alleles. Okay, so again, p is representing the dominant, so it's representing the A. So we're going to be talking about the number of A alleles divided by the total number of alleles, right? So we're going to have to add them in the denominator and make sure you include the division sign. Otherwise, you'll forget like I did. And then you click enter. And then over here, you're going to want to find the percentage of the B alleles. So you're going to do the B value divided by the sum of the total alleles. And click enter. And again, these two should add up to 1. So if you remember P plus Q equals 1, so if you kind of need to double check, you can always double check right here. And you should always get a 1. But you don't need to keep that because that's just double checking. Okay, so you found your P and your Q, and the reason why the label is over here says allele frequency for next generation is because, yeah, this is what would technically go. P and Q here are the same thing as this P and Q here. And if you're going to drag another generation, you would just scoot on over and put the P's and Q's up here. Again, you wouldn't be typing them, right? Because these will be changing. So you would click equals, you click equals H28 or equals J28 as your next P and Q value up here. So let me just show you what that would look like. You would go P equals, and then over here you would write equals, and then you would just simply click on this box and then click enter, and you'd have the value copied up here. So it automatically copies, so you don't have to worry about actually being accurate with the value. And it is up to you whether or not you want to click equals 1 minus this, or similarly, you can always just click equals and then click on this box, and it'll be the same exact thing. But that one becomes up to you whenever you do it. And then you would just be starting another version of this again. And you will be doing that. But right now, all I need is for this part to be completed. Now, the last part for each generation you have to do is to graph the genotype frequency in the next generation. Okay, so that's going to be based on these 3 values, right? Because it's genotypes. Okay, so we are going to be graphing these 3. I hate graphing on this. Okay, so right now, technically, I think this is accurate. Let's see how can I move this? Move this out of the way so you can actually see the numbers that we're referring to. So we're referring to 0, 5, and 12 right now. So the 0, obviously you can't see the 5 and 12. But this is really hard to read because there is no title, there is no axes labels. So we're going to go through and you're going to have to customize, and you're going to have to add axes labels. You're going to have to add a vertical axis label. You're going to have to add a chart title. Let's see. You can type in a title. So this would be P generation genotype frequencies. And so for each generation, you'd be able to say F1, F2, F3 after this, right? So that's our chart title. Our series is correct right now. Do we have a legend? We do have a legend. So you're going to, we can write none technically if we have our horizontal axis labeled. Let's see if I remember how to add in the axis label. Okay, the series is correct, but I need to add the x-axis. Huh. Okay, at this point, if you are at this point and you know better than me how to add in the titles and stuff, you can go ahead and tinker with that and figure it out. And then screenshot basically all of your work with the graph out of the way. Right, you move the graph out of the way and screenshot all of your work to show me that you were able to get to this step and submit it. If you want to kind of wait with me and figure out what's going on and try to add it, because I don't remember from doing it, then you can also wait and do that too. Why would it use row 22 as headers? Interesting. So you have to add the axis title, obviously, and that's going to be type. And our vertical axis title is going to be individuals. Because it's not frequency, because it's actual numbers, right? And then we're going to want our this isn't really the issue at hand right now. We just want to have an axis. I really need a legend. I honestly can't remember how to add these. Oh, how nice.